Hi, welcome everyone. Today I'm going to go over our 7th lab, which is all on molecular genetics, and more specifically, how you take a piece of DNA and you code it into a piece of messenger RNA and then ultimately make a protein from that. These two processes are called transcription and translation, and it's something that we also refer to as the central dogma. So let's go ahead and get started. So in this activity, you're going to be able to take a piece of DNA and you're going to code it into mRNA, and then you're going to code it into a protein. And like I was just saying, this process is sometimes referred to as protein synthesis or the central dogma. When you're discussing this flow of genetic information, it's really important to recognize the outcome. And proteins are super important in our body. They pretty much code for all of our genetic traits. So it's really important that there aren't any mistakes in this process. Now, before you dive into what is transcription and what is translation, there's different types of RNA that are involved. Now, remember, RNA is genetic information. It looks similar to DNA. It carries the code, but it is single-stranded and it performs a variety of tasks. So the first type of RNA that we're going to talk about that is extremely important in this process is called mRNA. And mRNA, well, the M stands for messenger. So what this RNA will do is it carries the message from DNA to a ribosome. The second type of RNA that you're going to come across in this transcription and translation lab is called tRNA. And the T stands for transfer RNA. And this is because what tRNA are doing is they bind up with mRNA in the process of translation, and they're carrying an amino acid. And they will transfer that amino acid to another amino acid to make our protein chain. Remember, proteins are just a long chain of amino acids that get folded. So when we talk about amino acids, if you put a whole bunch of those together, you get a protein. There are two steps in making a protein. The first step is called transcription, and the second step is called translation. In transcription, what the cell does is it takes a sequence of DNA and codes it into a message. And you might be asking yourself, well, why does the cell do this? Well, it does this because it doesn't want to take the DNA out of the nucleus. If you take the DNA out of the nucleus, there are increased chances of mutations or something happening to that DNA. So you want to keep it in the nucleus. So you need that message to go out. So what the cell does is it codes it into a message, and we call that messenger RNA. The messenger RNA will leave the nucleus and it goes to a ribosome. And at the ribosome, it gets translated into a protein. And that's with the use of tRNA. Okay, so in transcription, you're taking a piece of DNA, a gene, and you're coding it into a message, messenger RNA. Okay, and you probably learned in regular bio that DNA has 4 nitrogen bases: adenine, thymine, cytosine, and guanine. Okay. However, when you go to make your mRNA molecule, it's important to remember that mRNA does not have the nitrogen base thymine. Instead, it has uracil. So if you have an A nitrogen base on DNA, in RNA, it's going to get coded for as a U. That's what complements the A. So C will still go with G. That's the normal complementary base pairing. But A will go with U on mRNA. However, on DNA, if you have the base thymine, it will still code with A on mRNA because RNA still contains the nitrogen base adenine. So just remember, DNA has the 4 nitrogen bases A, T, C, and G, and A always goes with T and C always goes with G. However, in RNA, they only have 4 nitrogen bases, but it's adenine, uracil, cytosine, and guanine. So your lab this week is composed of 2 parts, part 1 and part 2. Part 1 is simply watching a tutorial by this biology professor named Mr. Anderson. And all I want you to do is to go to this website, watch the video tutorial to get you more comfortable about the steps in transcription and translation, and simply answer the questions on the video guide. In part 2 of the activity, you are going to be drawing a cat. I know that seems really weird, but I'm giving you a DNA sequence, and you get to choose. You can either choose DNA sequence 1 or DNA sequence 2. They are on the paper. So choose one of those, and then what you're going to do is take the DNA and you're going to transcribe it into mRNA. And remember the complementary base pairing rules. So if there's C on DNA, the complementary side on RNA is going to be G, and vice versa. G goes with C. The tricky part is when there is a base of thymine, T, on DNA, then A will complement it on RNA. But remember, RNA does not have the nitrogen base thymine. It has uracil instead. So when you have an A on the DNA side, you have to put a U on the RNA side. And I'll go over and give you an example in a second. So you're going to transcribe the DNA into mRNA. And then with that mRNA strand, you're going to translate it into what amino acids would be coded for at the ribosome. Figure out what the amino acids code for. So there, I'm going to show you a cat trait table, and these amino acids are going to tell you what trait is going to exist on the cat. And then when you have found all 6 traits that are supposed to be in that cat, then you're going to go ahead and draw your cat with those traits, and of course color and label them. Take a picture of your cat and insert it into the lab document. So here is a picture of one of the DNA sequences. So this is DNA sequence number 1, and you can see there's a very long DNA sequence there. So then what I said is you're going to take that DNA and you're going to transcribe it into mRNA. So there's the example down at the bottom left. You can see that the first DNA sequence is TAC, and then the second is ATA, right? So when you transcribe that into mRNA, T will complement A complements U, C complements G, A complements U, T complements A complements U. So your mRNA is going to read AUG, UAU. So then what you have to do is figure out, well, what do those 3 nitrogen bases represent? What amino acid are they representing? So you're going to use that chart that you saw in the Mr. Anderson video, and I also supplied it again here in the document, but you got to figure out what AUG codes for. And so if you notice, it's the start codon, and the start codon is methionine. Okay, so then what is UAU? Well, if you look on your table, you're going to see UAU codes for TYR, which is tyrosine, which is another amino acid. So you're going to keep doing that for all of the DNA sequence so that you have all the amino acids translated, so you can figure out what is the trait, or what traits are your cat going to have. So after you have translated and you've gotten all of the amino acids, you're going to come look at this cat trait chart. And this cat trait chart will tell you what your cat's going to look like. So for example, if you take a look at the first amino acid sequence, say you decoded your DNA and it read MET, TYR, CYS, GLN, GLU, ILE, and ASP. Well, that means that sequence right there is going to code for gray fur for your cat. So when you go to draw your cat, make sure that you put gray fur on your cat, and then of course label it with gray fur. Like I said before, your cat is going to have 6 traits, okay? 6 traits. So you know you'll be done when you have found 6 traits in your DNA sequence. Okay, good luck everyone. Make sure you take pictures of your cat, submit them into the document, as well as all your DNA and mRNA and amino acid translation, transcription stuff, and submit it to Blackboard by the Monday at 5:30. All right, bye everyone.